Neurons are nerve cells found in the nervous system, which includes the brain and spinal cord. Together, they comprise the central nervous system and ganglia of the peripheral nervous system. Neurons are specialized cells designed to stimulate other cells in the body by transmitting a signal. These cells are excitable, which means they function by using electrical stimulation. Through this electrical message, known as an action potential, neurons are able to initiate actions in the cells they target. These signals between neurons occur via synapses, specialized connections with other cells. These synapses allow multiple neurons to connect to each other to form neural networks. A typical neuron has 4 distinct parts. The first part is the cell body, the soma. This is the metabolic control center of the neuron, which is responsible for protein synthesis. The second and third parts are processes, structures that extend away from the cell body. They are responsible for conducting the signals to flow to or away from the cell body. Incoming signals from other neurons are typically received through its dendrites. Ongoing signals to other neurons flow along its axon. A neuron may have many thousands of dendrites, but it will have only one axon. The fourth distinct part of a neuron lies at the end of the axon terminals. These are the structures that contain neurotransmitters. Neurotransmitters are the chemical medium through which signals flow from one neuron to the next at chemical synapses. Neurons come in 3 functional classes. These classes are derived from the amount of cell extensions that are found on the cell body or the soma. Unipolar neurons have 1 extension connected to the soma. Bipolar neurons have 2 projections, and multipolar neurons have many cell extensions, including dendrites, and typically 1 axon. It is through membrane processes that neurons communicate with one another and also turn on, off, or regulate cell genome activity. In addition, membranes allow the organized and controlled movement of nutrients into and waste products out of nerve cells. Many of these membrane functions require the input of cellular metabolic energy, which accounts for nerve cell membranes, especially synaptic membranes, being the highest consumers of metabolic energy in the brain. 2 major membrane molecules are lipids and proteins, with phospholipids and cholesterol being the major lipid components in neurons. Membrane proteins give rise to molecular clusters which function as membrane receptors, channels, and enzymes that convert proteins and lipids into smaller or larger structures. These proteins are embedded in membrane lipids. Therefore, alterations in the structure and molecular dynamics of the membrane lipids will alter the dynamics, structure, and physiological function of the membrane protein. Such alterations have been implicated in neurodegenerative disorders like Alzheimer's and schizophrenia. Neurons can respond to stimuli and conduct impulses because a membrane potential is established across the cell membrane. In other words, there is an unequal distribution of ions on the 2 sides of the cell membrane. This is called a resting membrane potential, polarization. Once stimulated, neurons send electrical signals along axons. This mechanism, called conduction, is how the cell body of a neuron communicates with its own terminals via the axon. Communication between neurons is achieved at synapses by the processes of neurotransmission. 2 ions are responsible, sodium and potassium. An unequal distribution of these 2 ions occurs on the 2 sides of a nerve cell membrane because active transport proteins establish a gradient with higher sodium concentrations outside the membrane and higher potassium concentrations inside. During an action potential, sodium channels open, causing a rapid increase in the membrane voltage relative to the outside. Once a certain height of voltage is reached, the sodium channels close and potassium channels open to reestablish a low relative voltage inside the membrane. Once this repolarization occurs, active transport proteins reestablish the correct ion concentrations and ready for another action potential. The movement of an action potential across the nerve cell membrane is known as impulse. This is the propagation of the signal down the axon in a wave-like fashion. The speed of this impulse is influenced by the presence of myelin sheath, which are membranous extensions that sheathe the axons in successive layers and are produced by glial cells. This myelination is not shown here for animation of the impulse. The end of an axon leads to a synaptic knob, which has synaptic vesicles that contain neurotransmitters. Between the end bulb and the dendrite of the postsynaptic neuron, there is a gap commonly referred to as the synaptic cleft. So, pre- and postsynaptic membranes are not actually in contact. This means that the impulse cannot be transmitted directly. Rather, the impulse is transmitted by the release of chemicals called neurotransmitters. When an impulse arrives at the end bulb membrane becomes more permeable to calcium. Calcium diffuses into the end bulb and activates enzymes that cause the synaptic vesicles to move toward the synaptic cleft. Some vesicles fuse with the membrane and release their neurotransmitter through exocytosis. The neurotransmitter molecules diffuse across the cleft and fit into receptor sites in the postsynaptic membrane. When these sites are filled, sodium channels open and permit an inward diffusion of sodium ions. This causes the membrane potential to become less negative and approach the threshold for an action potential. If enough neurotransmitter is released and enough sodium channels are opened, then the membrane potential will reach threshold, an action potential occurs, and spreads along the membrane of the postsynaptic neuron. If insufficient neurotransmitter is released and the threshold voltage is not achieved, the impulse will not be transmitted through the postsynaptic neuron.